also one of the leaders of the Restore Consortium, and she's going to talk to us about using next-generation Tregs to reshape the immune balance.
Yeah, dear Chairman, ladies and gentlemen, thank you so much for giving me the possibility to guide you a little bit through our philosophy with respect to regulatory T cells, the other source of T cells. So why this is so important? Because in contrast to the effector T cells, manipulated genetically or not, we have to face another important issue in treating our patient, and this is a high prevalence of immune disease, which covers more or less 10% of the whole population and has an enormous socioeconomic burden. We have to pay in Europe annually more than 100 billion euro. Only for treating the patient to control the dominant immune pathology. And we believe, and I will show you a few data, that by using regulatory T cells, here, we can shift the immune pathology, the dominant immune pathology, towards regulation and hopefully also tolerance. And our strategy is to use the so-called natural regulatory T cells, the thymus-derived one, as a master cell product for any kind of product manipulation to broaden up the application for transplantation, autoimmunity, autoinflammation, as well as the RegMed field. Now, a few years ago, we start with our so-called first generation regulatory T cell product, what we produced from peripheral blood, only a small amount, roughly 40 to 50 ml, taken 2 weeks before transplantation. We select the cells of interest, enrich them, expand them for the first in human clinical trial to infuse at day 7 in a dose escalation 3+3 design, either 0.5 or 1 million or 3 million per kilogram body weight for checking the safety of this approach, as well as to get hints for any kind of efficacy. So this is a short summary of what we can state after an observation period of these patients longer than 3 years here. We can state in general that this approach is very safe. We do not observe any kind of infection and malignancies so far, and we have hints of efficacy, which means in comparison to the so-called standard of care, we have an excellent graft survival and graft function, despite the patients receive only one drug starting at the middle of the first year after initiating the transplantation. But everything is related to a but. We have to face 3 or 4 problems. One point is that we have to realize that we have only a temporary expansion of the regulatory T cells in vivo in the patient. The second important issue is that we do not see any kind of dose relation in respect to the clinical effect. And the next point is, even if it is a big step forward in transplant medicine to have an excellent graft function with only one immunosuppressive drug long-lastingly in place, we do not achieve any hints for tolerance induction, neither by clinical features nor by molecular signatures. And 3 of the patients have to be switched back to the standard immunosuppression, 2 because of rejection in the first year after transplantation, and the third one due to reoccurrence of the disease origin. This means we have to think how we can improve our product by different ways. And here comes the so-called European Consortium Reshape in place, which focus on 4 important issues. One is related to the manufacturing process and the modification of the product based on higher purity, specificity, stability, and functionality, which means in summary that we have to have an in-depth product analysis. Secondly, we are facing the point that our preclinical test systems are more or less not good enough to predict in vivo as well as in vivo outcomes and targets. And here, yesterday, you heard a lot of Patient on the Ship given by Uwe Marx. And we are thinking how we can improve the engraftment of the cells which we are manufacture. And everything has to be accompanied by health technology assessment, because my previous speaker already highlighted the cost of goods and the pricing and the fact that all therapies has to be available at the end to patient in need. So coming to the first point, enhancing the purity. I already highlighted that we start from peripheral blood, which means we start with a very, very low number of regulatory T cells, only 1 to 2%. And we could enrich them in the process. So we established up to 60 to 70%. And by the expansion strategy, we achieve 1,000-fold or 10,000-fold expansion. We have purity which range stable, independent on the patient characteristic by 90 to 96% after a production process of 23, and now we shorten it to 15 days. But if we want to manipulate genetically the cells, so we have to face one important issue, and this is that we manipulate also contaminating effector T cells. If we do it early, then the contamination of the cell population, effector cell population, is higher, likely, but it is also likely in the late manipulation, which means we need high purity at the early time point. And here we have, let's say, one solution. One solution is that we have cell sorting strategies in place. 2 systems are in place so far. One is the influx system, and we are working with the Taito. And here I show you that we could enrich the purity to 99% in the starting population. So the next point is redirecting specificity. In principle, we have 2 possibilities. One is that we select in the polyclonal repertoire the low frequency of target antigen-specific regulatory T cells. This is possible in the transplant setting. It is nearly Unlikely in case of autoimmune diseases, we can select them as the right one, expand them. The other point is that we redirect all cells either with transgenic TCRs or with CAR, you heard about. And we want to start and initiate in collaboration with Sangamo and Nantes, beginning of next year, the first in human trial using alloreactive HLA-A2 directed CARs. But we have to face different challenges. One is the appropriate target antigen. The homing at anyhow is a problem, which is important for tolerance induction. The in vivo stability of regulatory T-cell function and the safety issue. But on one important issue is also to address the question, do we need the same or different CAR cell CAR signaling for our CAR Tregs in comparison to what we know from the field of anti-cancer therapies. Last but not least, improve the stability and functionality, because we have to face three bottlenecks. One is the immunosuppression of the patient. The second is that the cells come in an unfriendly, inflamed environment, and they miss IL-2. All these impact the functional stability, and we work on several strategies to overcome these bottlenecks. One is that we create CRISPR-Cas-mediated CNI-resistant regulatory T cells. We want to start next year with this trial. For the inflammation, in principle, three possibilities are existing. One is the epigenetic editing of the regulatory T cells. The other is by using CRISPR-Cas mediated knockouts of the switch points. Here we focus on IL-6 receptor, and the last possibility is to stabilize the metabolism of the regulatory T cells by overexpressing dedicated genes and gene function. The lack of IL-2 can be overcome by delivery systems, either systemically, which is already done and proven in some proof of concept trials. So we are focusing on local delivery. These are the trials which are upcoming. I already highlight the A2 specific CARs, the CRISPR-Cas CNI, and the depleting strategy to have a better space for growing of the regulatory T cells. Now sum up, we have extensive experience in GMP manufacturing. We have a pipeline in place for next generation product with enhanced stability, specificity, and engraftment capacity, as well as we are working on adjuvant co-medication, which make the efficiency higher of our treatment strategies, and everything is related to preclinical studies in vivo and in vitro models, as well as health technology assessment. This is my acknowledgement not only to my team and the preclinical team and the clinical teams, also to the patient, and of course to the founders of different grants, the Federal Ministry of Education and Research, and the several grants given by the European Union. Thank you for your attention.
Thank you very much, Petra. Unfortunately, we don't have time for questions, so we'll need to move on to the next speaker.
Okay. The next presenter is Mario Assenmacher, hopefully I pronounce it correctly, from Miltenyi. And he will give his experience how to move from a supplier of the great machinery that they have to become a player in initiating company studies.
Yeah, thank you very much for the opportunity to present some of our results here. And as introduced already, I will dig a little bit into the clinical translation we are pursuing at Miltenyi Biotech. I think the CAR therapy has already been introduced nicely to you. So everything started with innovation 30 years ago by Selig Escher from Israel. Then there was a good example of a translation. Many centers in the US were heavily involved in this, Carl June particularly. And then finally, 2017, the first clinical products became approved. So these are the first generation products, CD19 CARs against B-cell malignancies from Novartis and Kite. But as any first generation products, there we are also facing good results, but also limitations. And I will come back to this in a second. So I think further improvement is possible and needed, of course, on all three levels, which means from efficacy point of view, reducing the amount of relapse, safety point of view, CRS, neurotox, as well as also already introduced to you from a point of view of broad application to really bring it to many patients and the high intention to treat success. The scope for Miltenyi is, as mentioned, we of course want to enable this from a platform perspective to allow academia and industry to apply this cellular therapy approaches. We want to bring innovation to this from both ends, from the platform end as well as from the CAR end. And we are doing clinical development of selected CARs. So this is a short overview of the ongoing CIT activities from Miltenyi. So we started with CD20 and CD19 CARs in B-cell malignancies, and as well as CD20 in melanoma. In all cases, first patient cohorts are under treatment. And then extended recently this to a tandem CAR, and I will come back to these two ones in a minute. So the first example, the CD19 CAR, from a CAR perspective, is reasonably conventional, but I think this is mainly tackling the challenge of broad application. So the concept of this clinical trial, which is running in Germany, is that we have not only multiple clinical centers from pediatric as well as adult side, but also multiple manufacturing centers. So this is an example where we try to establish proof of principle for a point of care, in a joint point of care manufacturing by multiple manufacturing centers. The core of this is the cell factory, which we established in Bergisch Gladbach in our headquarters, which serves with blueprints for the other manufacturing centers to make transfer and alignment of different manufacturing sites smooth and reliable. So this is a view into this cell factory, which is based on an innovative ballroom concept. So where multiple products can be manufactured in one Class C room in parallel, even for different products. So CD19, CD20, or the tandem in the same room at the same time. And this has been inspected by local authorities and the Paul-Ehrlich Institute. And we early last year, got a generic manufacturing permission based on this highly automated closed system for the manufacturing of different CAR T-cell products in this environment. So this should enable a decentral manufacturing of cellular products. First, of course, to foster clinical research and translation, but we also believe in this as an innovative model for patient care down the road. With this, I would come to the second example, which is more tackling the aspects of efficacy and bringing further innovation to the existing CAR T-cell approaches. So the 2019 tandem CAR application. So one issue we are facing with current CD19 CARs is also antigen escape, as for many monotherapies. For example, here are some data from the lymphoma side, where a significant portion of patients with disease progression show loss of antigen, of the target antigen. This is CD19 treated patients, which initially expressed both, but after therapy lost the CD19 and only remains with CD20. So to improve on this, multi-antigen targeting would be the right way to try to approach this. And one solution to this is so-called tandem CAR approach, which means that we have a CAR with two binding moieties. We generated this CAR, first CD20 binding and then CD19, both on a typical CAR backbone with CD8, 4-1BB and CD3 zeta. Of course, initially we established the in vitro preclinical data showing that this tandem CAR T cells, of course, can kill Raji tumor cells, which express both antigens, but they also are highly efficient in killing cells which only express CD19 or only CD20. This CAR T cells were also shown in the mouse model to work in vivo. So these are different days. After 1 week of tumor inoculation, the CAR T cells were transferred and compared to single CAR positive cells, and maybe focus on day 32. So the tandem CAR nicely cleared the Raji tumor cells from this mouse, very comparable to CD19 and CD20 single CARs. We challenged this even further in a high tumor cell model where the tumor had time for 12 days to grow up. And again, the 2019 CAR was able to control this high tumor burden. CD19 only had a harder time to do so. Combinations, a mixture of CD19 and CD20 CAR T cells is also possible to do so, but we, in this mouse model, at least saw higher toxicity, so lost some mice with this combination and not with the tandem CAR. This also has been moved into clinical in a collaboration with the Medical College of Wisconsin. Nirav Shah presented the first data end of last year at ASH, where he treated different lymphoma, non-Hodgkin lymphoma situations with this tandem CAR, starting from a cell dose of 2.5 times 10 to the 5, then now in a 2.5 times 10 to the 6 target cell dose. In many cases, fresh product, because also, I didn't mention this before, all of our CAR clinical trials are based on fresh products. And in this situation, we saw, let's say, expected toxicity, low level of toxicity, not more than expected. And also already, even in low cell numbers, responses. With the target cell dose, this is increasing. And also notably, patients being only, for example, CD19 respond well. Only one patient here, in this case, CD20 only, but at least shows also partial response. So first indications that this can cope with both target populations. Is this the end of innovation? I think, of course, especially when considering solid cancer, we have to take further steps to tackle not only multi-antigens, but also on the cancer itself, on the microenvironment. We probably need a higher level of control. And there are further technologies introduced, I think, already yesterday, which might be well suited to tackle these challenges we are facing towards solid cancer. Thank you very much for your attention.
Thank you. We have room for one question because you did make it on time. So one question, please.
Thanks. First of all, thanks for your talk. It was very, very interesting. Also, the model that you are proposing, I think that a lot of people will be interested in exploring this path. I have a couple of questions, clarification that I would like to ask you. The first one is about the central immunomonitoring lab that you were presenting in one of your slides, where you have decentralized IMPD manufacturing, but a centralized lab. Is that one only for the QC, for the release of the drug product, or for the long-term follow-up of the patient? And the second question is, all the deslocalized IMP manufacturing have been all inspected by PEI?
Yeah, maybe
Thanks.
to the first question, very good questions. I think, of course, the IPCQC is done locally at our cell factory as well as with the other manufacturing centers. So we established also a highly automated flow cytometry-based analytical procedures for transfection efficiency, cellular composition, and they are really running in the same manufacturing facility as the cellular product. So the immune monitoring central lab is only for following samples from the patients upon the clinical trial in a more centralized way. So clearly our vision is that, and it needs to be, that this IPCQC is very closely connected to the manufacturing, and to a similar degree needs a high degree of automation and standardization, which we are supporting with our platform. Second question was, yes, so the other ones, yeah, this is of course already some probably, I would say, disadvantages we are facing. For example, in Germany, this is typically done by the regional authorities. So they all need inspection by the regional authorities. This is typically backed with some guidance from the PEI. But we at least face heterogeneity, so that in the different regions, the authorities look slightly different onto this. So this is at least something, I think, where improvement is desired from this perspective.
Thank you very much. And we'll move to the next presentation.
Thank you very much.
Thank you. Okay, so I'd now like to invite Raquel Ophir to the stage to complete this session. She's VP of Research and Intellectual Property from Pluristem, and she's going to describe the development of an allogeneic placental-derived cell therapy from bench to bedside.
Okay, so I get the honor to stand between you and the coffee break, so I'll do my best to do it interesting and short. Okay, so 2 words about Pluristem, the company. It's an Israeli-based cell therapy company. Our cells are placenta-derived. I don't see it, but from full-term placentas. We expand them in our unique bioreactors under 3-dimensional conditions. The product that is achieved following this expansion is an allogeneic off-the-shelf product ready to be used. The administration is IM, local administration, and the mode of action is adaptive slow release of cytokines based on signals received from the patient's body, and to generate a long regenerative effect. So we are currently running a few clinical trials. 2 of them are pivotal phase 3 clinical trials for critical ischemia and hip structure, and additional clinical trials for various indications. We have 2 products, PLX-PAD, which is of the maternal side of the placenta, and PLX-R18, which is of the fetal portion of the placenta. In my presentation today, I will focus on the clinical development of PLX-PAD for critical limb ischemia. As I said, we're in phase 3 clinical trial for this indication. So just a few words briefly about the disease, peripheral artery disease. It is a chronic disease that progressively restricts the blood flow to the limbs. It is often associated with other comorbidities such as hypertension, cardiovascular disease, and etc. Critical limb ischemia is the end stage of peripheral artery disease, and it often leads to amputations and death. Critical limb ischemia and peripheral artery disease in general is a very underappreciated and undertreated medical condition. So I want to say a few words about the development of a new medication. You have this great idea in the lab and you want to make it into a selling product that many patients can benefit from it. So this path is quite a long path, and I must say it can be very specific at times. So let's start with what is needed in order to start this process. You need a good source of cells. We talked yesterday, Ornella talked about the placenta as a source of cells, and we're big believers in the placenta as a source of cells. These are ethically accepted cells, are rich and diverse. They have pro-angiogenic and immunomodulatory properties. The donors are young by definition, and it is an unlimited source due to birth all over the world. So once you have the cells, you need to know who they are, what they do. The characterization of the cells has been done, and these are the PLX-PADR mesenchymal cells in nature. They express positive and negative MSC markers. They have intermediate levels of HLA class 1 and of course no HLA class 2, but they lack to differentiate to the mesodermal lineages, which is what MSCs typically do. And this is why we believe that the mechanism of action, and we have evidence for this by now, is a paracrine endocrine mechanism of action. We can also see here that when the cells are injected intramuscularly into mice, they remain at the site of injection and gradually disappear, also supporting the endocrine paracrine mechanism of action. And indeed, over the years, we've intensively studied the mechanism of the cells, how they react in response to several disease signals, the immunomodulatory effect in response to inflammation, the effect to reduce oxidative stress signals, the effect to support muscle regeneration, and in specific related to critical limb ischemia, the effect to induce angiogenesis under ischemic conditions. Once we have identified and characterized the cells and have been able to identify the mechanism of action, we need to have a potency assay, biological assay that will enable us to release batches that have the same level of biological activity each time. This is an allogeneic product. We have to have batch-to-batch consistency. And indeed, we developed a potency assay that is related on the ability of the cells secretion to induce endothelial cell proliferation, in addition, a panel of angiogenic cytokines that are released by the cells. You also need to have your animal models to prove the efficacy and safety. The animal model that we used to prove efficacy was a mouse model of hind limb ischemia, where we could show that injection of the cells in this model, which mimics the obstruction of the blood flow to the limbs, as in the CLI patients, is regenerated, and we can see the blood perfusion following injection of the cells. Okay. I will skip this one for the sake of time, but we could also show the effect of the cells on migration and proliferation of fibroblasts, even if they are from young donors, or as is the case in our patients of old patients. In parallel to all the scientific parts, and this was discussed yesterday by Zami, you have to develop all the technological aspects of the cell product. You have to have a manufacturing capabilities. You have to have full quality control over the product and over the process. You have to control your storage, your shipment into the sites where shipping cells worldwide. The cold chain has to be fully controlled. And even the thawing of the cells at the clinical site has to be controlled. We wouldn't want to do all this work and then lose potency of the cells at the clinical site because the cells were left too long in a warm water bath. And based on all of this, which was submitted to all of these regulatory agencies, the EMA, FDA, Japanese, Korean, and Israeli authorities, we were approved to enter into a phase 1 clinical trials. So phase 1 clinical trial, as you probably all know, you're supposed to, the aim is to show the safety of the cells, but of course, for us as the developer of the cellular product, we want to see some hints of efficacy as well. We have seen in our 2 phase 1 clinical trials improvement in the oxygenation of the skin, as well as reduction in amputation rate and death, and also, interestingly, closure of the wounds, which are typical to these patients. This leads us to a phase 2 clinical trial. In the phase 2 clinical trial, we wanted to look at different aspects of the cells, especially dosing regimen. We looked at 2 different doses in different dosing regimens, 150 million cells, 300 cells given once, and 300 cells given in 2 administrations, 3 months apart. What we could see that not all treatments worked the same. The best treatment was achieved when we gave 2 administrations of 300 million cells, and it was even better when it was given from 2 different placenta donors. This brings us to another very important point, which is the need to do a responders analysis. And this is something that we will need to do in all of our ATMP studies. We need to look at what are the results of the individual patients and try to correlate them to the treatment, to the dosing regimen, and to the characteristics of the patient at the time of administration. So to summarize, in order to reach the approved product, the long climb that we were talking about, we need to develop the non-clinical aspects, the quality aspects, clinical development, manufacturing, logistic, all leading to the pivotal clinical trial. And then once we start obtaining clinical data, we have to do a lot of analysis of the responders and try to maximize the effect and be able to choose the right patients in order to get a maximal effect. And I think this is one of the goals of the Restore here, to be able to join forces and be able to centralize a knowledge of things that are common to all of these developments, so that we can reach the summit in as many as possible ATMP products. I would like to thank the agencies that support us, the Israeli Innovation Authority and the Horizon 2020 under the consortium of the PACE, who is supporting this clinical development. This is the Pluristem team. I want to thank our collaborators in the PACE and Dieter and Petra for their involvement in this project. Thank you very much.
Thank you very much. In the interest of caffeine, we're not going to take questions, I'm afraid, but I'm sure any of the Pluristem team will be happy to discuss. I'd like to thank all of our speakers from this session very much for their really interesting presentations, and I'd like to close this session. Thank you.